So in this video, we're simply going to talk about enzymology, which is just the study of enzymes. And we're going to begin the discussion by first describing what enzymes are. So what are they and what do they do? So the majority of enzymes that we're going to be having are protein in nature, though some of them are going to be in form of RNA molecules, or rather DNA molecules that are translated to create proteins. We're just saying each human cell contains thousands of these enzymes, providing specific help throughout the body. So enzymes are important because they help us. They keep us alive and well. For example, they perform a necessary function for metabolism, and this is also sometimes applied in industrial applications, such as the foods that we take in, the drinks that we take. So enzymes are biological catalysts. They speed up our chemical reactions and they reduce the threshold minimum amount of energy needed for a reaction to start. That's activation energy. In short, they remove reaction blockers or barriers. That's the main purpose. That's what they do. Yeah, so we're saying these are protein in nature catalysts that are produced within living cells. That's what they are. What they do is simply what we've listed out here. And we can go in detail to talk about what they actually do. So in terms of illustrations, let me give you examples so that you understand the essence why we have to learn and understand this. So in digestion, so I'll just be listing and explaining. In the digestive system, enzymes help the body by breaking down large complex of molecules into smaller ones, such that the body is now going to be able to consume them or utilize them by getting the necessary things or nutrients that are needed to use as fuel. So that's the main thing we can talk about in terms of digestion. We can talk about in DNA replication. So each cell in the body contains DNA. That's just the information that makes up that cell. Let's take it to be the formal definition of DNA. Each time a cell divides, the cell needs to copy its DNA. Enzymes help in the process of unwinding the DNA coils that you have. So DNA is a complex structure. So under DNA, this I'm just going to say it helps. Enzymes help in DNA replication, which is just the preservation of genetic information from mother cells to daughter cells. Yeah, so the main thing you just have to take away from here is that enzymes, ligase in particular, help in the unwinding to unzip the DNA molecule so that it replicates itself. Yeah, we're going to talk about that later on under DNA transcription and translation. For now, we're just discussing enzymes. The other property that we can talk about is in terms of detoxification, which is just going to be written as liver enzymes. So the liver breaks down toxins in the body. To do this, it uses a range of enzymes to facilitate the whole process by destroying the toxins. Anything that is not needed by the body is rendered useless and has to be excreted out. So there are some enzymes that help in doing so. So these are some of the practical examples why we need enzymes in our living cells or in systems that make up the human body. That's what we have. So some other examples that I may just point out without fully explaining them is hormone production, cell regulation. We have the creating movement to make the muscle contract or relax, the whole transporting of materials around the cell. We need enzymes to do that. Cell respiration, we need enzymes for that process to happen as well. So now that we've discussed what enzymes are, we now have to talk about how they work. We know why we need enzymes now. So how do they work? That's the main idea that we need to understand. So in as much as we said enzymes are protein in nature, meaning there's a limit in terms of temperature. There are things that are obviously going to affect the rate at which these enzymes are going to be working. Now, let me correct that statement I said earlier on. So mostly when people think about enzymes, they think about anything that speeds up a chemical reaction. So there are cases, rare though, where enzymes actually slow down the chemical reaction. And slowing down a chemical reaction facilitates the reaction to occur in a smooth way. So now enzymes work in such a way that they can be reused. So these are not, they can be reused. They are not denatured, so we can just say do not get damaged in the reaction process. So maybe we must add that to the definition of a catalyst. So they can work over and over again. If you look at the first image on our here that we showed, we're just saying if we have a particular enzyme, an enzyme always has two sides. There's what we call the active site, there's what we call the allosteric site, the inactive site. So if you have a substrate like this guy here, substrate is simply the substance we are working on, the substrate that the enzyme is working on. So we have substrates that we are trying to combine or build. So we have a complex substrate that we're going to add. It's going to be attracted to the active site because they're going to have what we call a lock and key type of design. So the substrate comes there, the enzyme does the work, they go separate ways, right? So the faster this happens, the more products we're able to make because the active site is going to be untampered with, and we're able to attract another substrate and also make another product. Yeah, so if we were to talk about how enzymes work, the first thing that we can talk about is the lock and key model or design. So we're just saying the lock and key model simply tells us that every enzyme can only work on a particular substrate. So the substance that we are working on is called the substrate. Every substrate has a specific enzyme that can work on it. So this is basically what we mean by the lock and key model. And apart from the lock and key exact model, we have what you call the induced fit model. So in the induced fit model, we're just saying, this is a newer way, this is a new method, of course, and the active site is not exactly the best fit for the substrate. But in this model, the active site is going to change shape as it interacts with the substrate so that the substrate can now perfectly fit in. So the active site is going to be adjusting itself until it locks in the substrate. And that's when the catalysis is simply going to start. Yeah, so it's induced because it's partially forced a bit initially because the active site was not an exact fit for the substrate. So we have this new model that they've discovered, and this is going to be fully explained after we talk about certain factors that affect enzymes. So if we talk about examples of specific enzymes and their functions, so the first thing that we're going to talk about is temperature. We're saying the catalytic activity of enzymes requires an optimum temperature, and this is mostly going to be 37 degrees Celsius. That's the body temperature. Meaning enzymes will simply be vulnerable or they'll be destroyed after the 37 degrees Celsius limit has been exceeded. Yeah, so this change is going to cause a drop or a complete halt of the reaction. It's going to bring a stop to the whole chemical reaction between the catalysis of the enzyme on the substrate. And also, lower temperatures can also slow down the rate at which enzymes are going to be acting. Yeah, so temperature is important. We have to talk about temperature and also pH. Yeah, so here if I may add, there's pH. Enzymes are sensitive to changes in pH. As in temperature changes, extreme low or high gain lead to denaturation of these molecules. So the concentration of hydrogen ions affects the ionization of acidic and basic groups. So we're just saying pH has to be steady depending on the environment. So the pH in the stomach, for living cells that are in the stomach, they are used to work at that particular pH of the stomach surroundings. You can't use those same enzymes, let's say in the lungs or anywhere else. Yeah, so the pH matters. Yeah, this is important because some of the created enzymes in labs, we have to take into consideration the normal pH ranges that they're going to be having. Because once you change the location of an enzyme to an area where the pH is abnormal to what it's used to, it's simply not going to do the same work that it does at its original conditions. So that's what we have. Now these are some of the the metabolic sources. These are some of the sources of enzymes: metabolic enzymes, digestive enzymes. They're just explaining the same thing that I had said at the beginning, to say we have systems where these can actually be found, and we have these enzyme types. They are oxidoreductase. These guys that cause reduction and oxidation. They are transferase. These guys are responsible for everything that has to do in terms of the transfer of chemical groups between molecules, or within cells. Then we have hydrolysis. So you notice that the naming of the enzymes always ends in ASE. We have ligase. This is the guy that is responsible for the unzipping of the molecules, the DNA molecule, the unwinding. We have isomerases. This catalyzes the isomerization of molecules. They change the shape of a molecule without changing the molecular weight. Remember, we're still emphasizing the fact that enzymes are going to be spearheading the whole biochemical reaction, but they don't get changed in the process. That's basically what we have. So some of the examples are simply going to be, we have lipases. So we have these guys here. These are simply a group of enzymes that help in the digestion of fats in the gut. So these are lipases. Then we have the second one, we have amylase. So this is found in saliva. This helps the change of starch into sugars. So this breaks down starch to sugars that are easily digested. Then we have maltase. So maltase, this also occurs in the saliva and breaks the sugar maltose into glucose. Remember that two glucose molecules made up maltose under carbohydrates. Now we're saying this guy breaks down the big structure into the smaller structures. This is maltase. Then we have the other one. So these are the common ones that you'll be finding that are discussed the most when I'm looking at them. We have trypsin. These enzymes break down proteins down into amino acids in the small intestine. So these break down proteins back to amino acids. Yeah, so this is basically what we have. Then the fifth one, we can talk about lactase. Hope you remember what lactase does. So lactase breaks down lactose, the sugar in milk. It breaks it down into what? What happens, or what do we get when we break down lactose? What do we get? If you still remember, so here we'll just get glucose and galactose. That's basically what we're going to get. Yeah, then there is helicase. Yeah, so point of correction on the part I said ligase there when we talked about the unwinding of the DNA. The actual enzyme that is used for the unwinding of the DNA strand is simply helicase, to unzip it. Yeah, so helicase enzyme and reveal the DNA. It unwinds it because DNA is very much cold. You see when you talk about the structures of proteins. Then we have DNA polymerase. These enzymes synthesize DNA from deoxyribonucleotides. So we have DNA polymerase. So these terms, they will be more used when you talk about DNA replication, transcription, and translation. Yeah, so these are some of the examples of enzymes that you have to get yourself familiar with. This is what you have. Now, aside from this and talking about factors that affect enzymes, we talked about the three important ones that I need you to have on your fingertips. So factors that can affect enzymes, I'll re-emphasize. The first one we talked about is temperature. So they have an optimum temperature, meaning exceeding that particular temperature, the enzyme gets denatured. Then two, we have the pH. So the pH is important as well because every particular enzyme has a certain range of pH that it can work in. So let me just give you the diagrammatical effect of temperature. And also, okay, so let me give it to you here. So this is what we have. The enzyme activity is going to be affected, depends on the temperature that you have. So you see that the maximum, the optimal rate of the reaction is somewhere above 40. So 37 to 40, you are seeing the enzyme giving us its best type of reaction, its best activity. But as soon as we reach a certain limit, that's the optimal temperature, the enzyme gets denatured, it gets destroyed. The whole idea of a catalyst is that it shouldn't be destroyed in the chemical reaction process. Yeah, so if it gets destroyed, meaning we cannot reuse it. Yeah, so we need to observe, or rather know, the temperature limit of each particular enzyme that you're using in a chemical reaction. Okay, so apart from that, look at key feed model. Okay, so now we're going to look at certain terminologies that are going to be used. So what are some of the terms? So apart from this, we can also talk about inhibitors. We'll discuss these guys later on. That's what we're trying to do. Inhibitors, they can also affect the rate at which a chemical reaction is happening. Now, there are two terms that I need you to understand before we even proceed, and those two terms are simply cofactor and coenzyme. So whenever we discuss coenzymes or cofactors, so we're just going to say some enzymes cannot function unless they attach to a specific non-protein molecule known as a cofactor. So a cofactor is anything that attaches itself to an enzyme. That's a cofactor, right? Now, we have two types of cofactors. We have the actual cofactor, and we have what we call the coenzyme. So the cofactor, this can either be inorganic, that's what we are saying, tightly bound to the enzymes, helps active enzymes often as a prosthetic group. Then the coenzyme will attach directly on the active site. So these are always organic, loosely bound to enzymes, act as a carrier for chemical groups of electrons. So we're just saying the coenzyme is going to be attached to the active site to make the active site to be compatible with whatsoever substrate is coming, the incoming substrate. The guys that are simply cofactors, the ones who are saying they're ions, these are obviously inorganic that can loosely bond to the enzyme to ensure it can function. The coenzymes, they are going to be loosely bound to allow the enzyme to do its job. When a cofactor bonds tightly with an enzyme, it is known as a prosthetic group. So they can sometimes produce what we call inhibition. So this is what I want to talk about here. So to ensure that the body's systems work correctly, it's necessary sometimes to slow down the enzyme function. For example, if you say an enzyme is making too much product, right? You want to limit it a bit. You want to control the production rate. The body needs to do it in such a way such that it won't completely stop the whole production. And these are some of the things that we're going to talk about to inhibit. So the first thing that we're going to talk about are called competitive inhibitors. So these are competitive inhibitors. So from the word competitive, it simply means this guy is going to compete with a substrate. So this inhibitor molecule blocks the active site so that the substrate has to compete with the inhibitor to attach to the enzyme. So it limits the rate at which the substrate is going to be finding its way to the active site. So these compete with the substrate. These are what we call competitive inhibitors. They're going to have the same lock and key design like what the substrate and the enzyme has. Yeah, it will just reduce the number of substrates combining with your enzyme. Yeah, so these are called competitive inhibitors. Then the other one is non-competitive. So non-competitive just does the opposite. It will just bond to the inactive site of the enzyme, hence deforming the active site. So if you have, let's say this is your enzyme is designed to be like this. So let's say you have something that comes like that. This is your substrate, right? This is your substrate. I'll say S. This is our enzyme, the ES complex. So the active site can just come this side. Let's say our active site is, this is the inactive site. So our non-competitive inhibitor won't fight for the active site. It will just come on the other side. Let's say, you can even say it's a sphere like this. it will get attached to this guy, and the enzyme, let's say, will flip. Let's say it will be deformed for a bit. So to accommodate the substrate, the enzyme is simply now going to change, and now the enzyme looks a bit more like this. So it gets big, right? It gets deformed such that the substrate cannot perfectly fit, because this guy is, let's say, smaller than is now smaller than this. So this is what we call the non-competitive inhibitor, because it bonds on the other side. Then we have what we call the uncompetitive. So we have C, uncompetitive inhibitor. So this inhibitor binds to the enzyme and the substrate. The products leave the active site less easily, which slows the reaction. So the uncompetitive is simply going to bond to the enzyme substrate complex. So after the enzyme and the substrate have already bonded, so let's say we have something like this, they've already bonded, let's say they are already bonding. This non-uncompetitive inhibitor is simply going to bond, let's say, around this, so that we lessen the rate at which the product is going to be released. Because we know after the enzyme substrate complex, what is produced is simply the product, the desired product, and the enzyme, which is left unchanged. Yeah, so this is reversible because we can easily detach this to produce our product. Now we have the last type, which is called irreversible inhibitors. So these irreversible inhibitors are those type of inhibitors that can bond to the enzyme and permanently deactivate it. So these can actually damage the active site. The fourth one, we're just saying irreversible inhibitors. So these can bond with the active site, then destroy it until it cannot bond with anything else. That's basically what we have. So these are some of the terms you have to be okay with when it comes to enzymes. And aside from these inhibitors that we have other terms like apoenzyme. When you say apoenzyme, we just mean the inactive site. Yeah, so when you say apoenzyme, this just means the inactive site of the enzyme. That's what we mean. Then while holoenzyme, the active form of the enzyme, that includes both the apoenzyme and its necessary cofactor. So the apoenzyme, which is simply the inactive form of an enzyme, can actually be called, this is going to be holoenzyme, if there is a suitable cofactor that we can use to attach. So when you combine a cofactor and an apoenzyme, we simply produce a holoenzyme, because we're just going to make this now to change from being inactive, and now it becomes active. Yeah, so that's basically what we have. Hope this makes sense. So this is roughly all about it, that the things that you just have to know about enzymes, probably with a few illustrations from your Shihata notes, but this explains it all. These are just the relevant things you need to know. Yeah, we talked about this. There is so they give us the optimum temperature, pH, for each particular example of enzyme that they give us. So this is just re-emphasizing what we discussed in terms of the pH being very vital to the type of enzyme that you're dealing with. Yeah, so this brings us to the end of enzymes. Thank you so much for watching.